Hey, I'm Mr. Williamson, and this video is about DNA replication and how it might make mistakes. Before we get started, make sure you're taking good notes. I personally like outline, but I incorporate that into a Cornell style. But you do whatever gets your car to go forward. Make sure you're asking questions in the Edpuzzle or in the classroom. Let's get started. Here we go. So before this, we've talked about how DNA, the structure of DNA. We talked about what it had to do, what Watson and Crick knew it had to do. It had to be able to store information, it had to be able to copy, it had to be able to make mistakes, and it had to be able to be read. All right. In our last video, we talked about the structure of DNA, how a nucleotide consists of a phosphate, a sugar, and a nitrogenous base. And there are 4 nitrogenous bases in DNA: guanine, cytosine, thymine, and adenine. Okay. And adenine and thymine pair up, guanine and cytosine pair up. Crooked letter, straight letter, straight letter. Remember that? All right. Let's talk about how DNA can copy itself. First of all, remember that it's a double helix. What's on one side of the letter is complements what's on the other side of the letter. And I explained that in the previous video, where A's with T's, G's with C's, and they complement each other. What's on one side is not determined, but it complements what's on the other side. All right, and they're anti-parallel. One of in one side the sugars are pointing up, and the other side the sugars are pointing down. I'm going to explain that a little bit more detail. You don't need to worry about it. Just it'll explain some things that you're going as we go along. So let's take the following example. We have a short sequence of DNA here, just as an example. And you can see the A's and T's, G's and C's, et cetera, et cetera. Well, the first thing we need to do is unzip it. All right? We're going to use an enzyme called Helicase. No, you don't need to know that. I'm just giving you the name of the enzyme. The enzyme Helicase comes in and it breaks that DNA straight down the middle. It unzips it. It literally unzips it, very similar to a zipper. So you have the original DNA here. This is the original. And then we unzip it, and the helicase is moving that way, just like a zipper, and it unzips it. And so each one of these would be an A and a T, and on the other side would be a T and an A, et cetera, et cetera. All right? Helicase unzips the DNA. The next thing that comes along is based on our pairing rules. A's with T's, G's with C's. Straight line, crooked line, crooked line. All right? We just pair it in. You've unzipped the DNA. You've unzipped the DNA, and we just put it, this is an A, we're going to put a T there. This is a T, we're going to put an A there. And on the other side, it does the same thing, A and T. It works exactly the same based on our pairing rules. And what we end up with is 2 identical strands of DNA. And this is the original. There's the copied ones. And you can see, first of all, across the way, they are all the same. We've replicated it. And it matches the DNA we had originally. All right, there's the 2 strands. There's our original strand. We now have 2 exact copies of that DNA. So by using the side of the DNA, each side, as a template, we can pair up the bases. The T's goes with A's, the A's goes with T's, the G's and C's, and G's and C's. We can pair them up. And as a result, by using that template, because it came from the original one, we get the exact same thing. Okay, here's a diagram of a DNA, one of the replicating itself. And first of all, I just want to point out this is a good diagram because it's not just a twisted ladder, but there's a large groove and a little groove. All right. But you can see all we've done is unzipped it, and they're putting in an A with a T, and the next one's a G with a C, and et cetera, et cetera. All right. Another view of that same thing. We start out with the original. We unzip it. The helicase would fit in here, and I'm going to show you a better diagram, not a picture, but a diagram. We start bringing in the different pairs, matching up the complementary sides. And as it works its way up, we just keep going until we end up with 2 identical DNA molecules. Unzip it with the helicase. Again, you don't worry about the helicase. All right? By the way, there's an enzyme that puts this in. There's an enzyme that's called DNA polymerase. Don't worry about it. All right? Helicase unzips it. DNA polymerase is the one that pairs up the right bases. And it just works its way up the DNA molecule until you get two of them. This is a really accurate diagram. Diagram, again, not a picture, where you have the helicase here, all right, unzipping the DNA. You start, which is coming from, in this diagram, from a chromosome, just showing you that it is DNA, all right? And remember on the complementary parallel thing, the anti-parallel? The anti-parallel, don't way over your head. But if we look at the sugar, all right, five-sided, they number the carbons. And so there's 5 carbons, and this one might be like 5, and this one over here is 3. Well, on this side, it points the other direction. And this is 5, and this is 3. So we have, they can only, and I'm probably getting this wrong because you don't need to know it, and I don't know it. It can only work one way. It can only work from the 3 to the 5. Let's say it could be 5 to 3. I don't know. I don't care. You don't need to know it. Just showing you something that you might want to observe. All right. It works only from the 3 to 5. So this one, as it unzips, can work its way along the DNA the entire time. But on the other side, we can't because it's going backwards. All right, we can only fit it, we can only go from the 3 to the 5, so we can only work this way. So we have to wait until it unzips some. This will keep working its way down, and then a new DNA polymerase will come in and fill it in there, and a new one will come in. So the one side works, just unzip it, and it just starts going down the DNA. The other side has to unzip a little bit, fits in the DNA polymerase, and fills that part in, and then another DNA polymerase fills in the part that we just unzipped. And the other, so one side's undoing it, is copying it exactly the same, just going down the DNA strip. The other side is going the opposite direction, and it's doing it in sections. Do you need to know that? No. What do you need to know? You need to know that there's an enzyme that unzips the DNA. We match the A's and the T's, the G's and the C's together. And when we're done, we have 2 identical strands of DNA. So the question is, if I can copy it, how does it make mistakes? Well, once in a while, it doesn't quite work this way. The A's and the T's and G's and C's don't go together. A mistake is made. Normally, it's corrected. It's not a big deal. Happens all the time. Happens when you're exposed to ultraviolet light, when you're exposed to some chemicals, things like that are in smoke and things like that. All right. And normally in this diagram, it shows in this diagram, you can see where the G and the T don't match up. G's crooked line, straight line doesn't work. Straight line, crooked line don't work. So what happens? Well, there's an enzyme that comes in. It takes out this entire section of the DNA. It replaces it with a new strand that does match, and when the correction is made. But it doesn't always work that way. Once in a while, a mistake is made. These are 2 examples, and there are probably more. But these are 2 examples. Guanine, all right, does not pair with thymine. Straight line, crooked line. But for some reason, the 2 bonds that are here do pair up. Now, thymine has 2 bonds, and let's see, I've got to figure this. And guanine, I believe, has 3 bonds. Okay? So, yeah, guanine has 3 bonds. Cytosine has 3 bonds. So there should be another bond over here. There should be another bond on this one right here. But it didn't work out that way. Somehow the DNA forced it together. Kind of like maybe when you're doing a jigsaw puzzle and you just get really pissed and you just like, this looks like it fits, and you make it fit. And then later on, you're like, okay, that didn't work like I wanted it to, but you forced it in there, all right? It's like fitting a square peg into a round hole. It'll work, it doesn't work. It'll fit, it doesn't work, okay? So you have the DNA that should be repaired, but once in a while it's not. So the question is that really a big deal, all right? Well, it depends. It depends a lot on what the part of the DNA is. It could be the DNA, in this case, isn't red. If we're talking about a skin cell, and I'm going to use that as an example because we all go out in the sun and we're all exposed to ultraviolet light. and ultraviolet light damages the DNA. And normally your DNA fixes it, but once in a while it doesn't. All right? But if it's not a skin cell, if that part of the DNA, out of those 200 trillion base pairs, if we ruin the base that has codes for a liver cell, who cares? Doesn't affect your skin because we're talking about a skin cell. It's kind of like we're reading chapter 12 right now. I think it's chapter 12. Pretty sure it's chapter 12. We're reading chapter 12. If your little brother comes in and destroys chapter 5, what do you care? Other than you'll probably have to pay for the book. But you don't need to worry about it for studying because you're not on chapter 5. You're on chapter 12. So no big deal. You've damaged chapter 5. Chapter 12 is okay. It's no big deal. We damaged the part of the DNA that's a liver cell, but we're not talking about a liver cell. We're talking about a skin cell. All right? Or maybe it just, the DNA, it doesn't do anything. Maybe damaging it even in the skin cell just causes the whole cell to die. Well, you losing one cell is not a big deal. You probably do that when you scrape against the wall, turning the corner a little too close. You damage some cells, your body isn't going to suffer. But if we damage it in the right way, it can become bad. It can become something like cancer. All right? But where a cell just multiplies and divides and grows, divides and grows, divides and grows. It never becomes a skin cell. It just divides and grows, divides and grows. All right? So the question is, how can that one base pair that's damaged be such a big deal if we're talking about 200 trillion bases along the way? Well, that all really depends on how we read it. All right? How do we read DNA? That is the question that we need to answer. And I think that's what we're going to talk about in the next video. All right? So right now, so far, what we've talked about is the things that Watson Crick knew it had to do. It had to be able to be read, store information, copy itself, and make mistakes. We know about the structure. We know that adenines pair with thymines, guanines pair with cytosines, purines and pyrimidines together, purines and pyrimidines together. We know the structure of those bases. We know that a nucleotide is a sugar and a phosphate, and the backbone, and then a nitrogenous base, the A, the T, the G, and the C in DNA. All right, and we know how to copy it. DNA helicase comes in and unzips the DNA. DNA polymerase comes in and matches up the correct nucleotide with the corresponding one on the other side, using the original DNA, each side of the original DNA, as a template to put in the correct base. We know all that. What we don't know is how we can read it. And like I said, that's the next video. Again, I hope you took good notes. If you have any questions, ask them in Ed Puzzle or ask me in class. Good luck.